John, welcome.
Thank you.
Thanks for coming on to Beyond.
Oh, it's a pleasure.
Yeah, not a bad day, huh? Not a bad view?
No, it's beautiful sunset, nice weather. I couldn't complain.
Yeah, so you were doing some really groundbreaking, cool stuff as I read your background. And you're really on the cutting edge of genomic research focused on gene-based technologies to, in part, foil cancer before it can metastasize. And I remember I had Annette Walker on, who's with City of Hope, and they just brought this big cancer initiative in. So it's really a topic near and dear to my ears and what I'm following recently. You spent the last 10 years focused on halting cancer. Where did this passion come from?
Well, it's interesting because I was in wireless for a long time. I built the PacTel, PacBell, and Metro PCS networks out here in LA area. And a friend of mine was investing in a medical device company that did intraoperative radiation therapy. So they could radiate cancer during surgery. So I took over the company that we were working with and I ran it for 7 years, got it converted, it was a public company, took it private. And it got me very interested in the cancer space because what you start seeing is cancer is a very individual disease because it's very cell-based and no two cancers are alike. And what I was working on with radiation therapy was all on the back end. So we dealt with a lot of the worst cancers and really where people were in worst case,
Stage 4.
Yeah, stage 3 and 4 or early stage breast cancer. Yeah. And when I left that company, I started trying to figure out how can I impact cancer more on the front end. And what I saw working with some of my customers was that genomic research was really driving the front end. It made perfect sense because it's cell-based. Because what happens, you start looking at genomics, you start looking at cancer not by location. So you're not saying, oh, it's breast cancer or colorectal cancer. Mm-hmm. You start looking at by mutation. So when you look at the one that everybody knows is the BRCA1 and BRCA2 gene for breast cancer. Mm-hmm. BRCA1 and BRCA2 are also relevant in pancreatic and colorectal cancer. So you're not attacking breast cancer, you're attacking the BRCA mutation, you're attacking a PIK3 mutation. So you start attacking not only the mutation itself, but the pathways that it's going to take when it mutates, you get subclonal mutations.
Can you stop it in its tracks when you attack the mutation? Well, is that the end goal ultimately?
The whole goal in genomic research, and what we're seeing is it's really driving to combination therapy. And what combination therapy is, it's what they use for tuberculosis and what they use for AIDS. Because they realize you can do something that's going to really attack the cancer, which is a lot of what chemotherapy does. But then at the same time, you wind up hurting the patient with a really cytotoxic treatment. So when you look at it, you say, okay, what we're going to do is we're going to attack it from 3 fronts. We're going to attack the tumor itself. We're going to attack the pathways where it travels, because the scary thing about cancer is it mutates much faster than normal healthy cells. So you want to stop the pathways. So if there's a mutation, you can stop it and kill it before it mutates. And then you're also going to provide the immunal response. So when you look at immunotherapy, you're going to help your body produce anything that fights that cancer and that tumor. So you're usually using anywhere from 3 to 5 separate agents. And when I say agents, it's drugs, but it's not necessarily chemotherapy. It's hormonal therapy, it's immunotherapy, it is chemotherapy. But you're using it in much lower doses, and most of it is oral agents. So you can take them, it's a pill. So the beauty is you have very, very low toxicity, much healthier patients. So we've seen patients that we've treated with combination therapy at Avera McKinnon, basically we've taken out of hospice.
Wow. So because today with typical therapy, with chemotherapy, some people get really sick, they get radiation poisoning, and I know a lot of people I don't want the technical methods, the most advanced methods, and like Steve Jobs, they take other Eastern approaches to addressing cancer, which is not without risk. So what you're talking about is something, maybe it's not an in-betweener, but is much less egregious to the body than chemotherapy, and something that's more tolerable to fight the same disease.
Yeah, when you talk about, it's very personalized. So when people talk about precision medicine, personal medicine, that's really what this does. It tailors your treatment specifically to you, to your genetic makeup. So the interesting thing is, when you look at chemotherapy, the standard of care is we treat you with a chemo, basically blast with the chemo agent to hope that it kills the cancer. When it recurs, the next treatment is it treats you with a new chemo. And by the time you get to the third recurrence, the third chemo, your chances of winning the lottery at 292 million to 1
Is better than
3,000 times better than being cured on a third chemo agent. So that's why when you look at this, you really want to do the combination therapy because you can adjust it. You can treat, and that's what they do is you start doing what's known as a liquid biopsy. And what liquid biopsy does, it detects any cancer cells. You'll see it's circulating DNA. So you start looking at circulating tumor DNA. You'll hear people say ctDNA. That's what it is, because they'll see the tumor cells in the blood before the new tumor forms. So now they can change whatever they're giving you to address it before it gets too bad.
Now, is there ever going to be technology, in your opinion, where you can know that you're a potential candidate to get cancer, like way before it happened? Is that happening in blood tests, or is there a way, if I go to the doctor for a regular physical checkup, where they could say, hey, you got some of these markers and just be on the lookout, or we think you should maybe be aggressive in your care and treatment?
Yeah, there's panels now that call it predisposition. So they look at you and see if you're predisposed, and that's why people look at the BRCA mutation and say, well, I'm predisposed. The important thing is it should increase your surveillance. It's not a real call to action right away. A lot of people, panic and say, well, I need to get an oophorectomy and a double mastectomy. What it really should is it should provide you more surveillance so that if you're going to get it, you're more vigilant. Because caught early, cancer is being treated very well.
And surveillance is what, a blood test, or what does that look like exactly?
In many cases for breast cancer, they do mammograms. For a lot of other cancers, it's a blood test.
Okay. And pretty effective?
Very effective.
So you're the CEO of SimpleSeq?
Yes.
Did I get that right?
Yep.
And it's out of Marietta Genomics company, or it was formed, right?
Yep.
So talk to us about some of the setbacks. I mean, we can always talk about the glamorous part of being a CEO and take a company from public to private and doing great things, but talk to the audience about some of the setbacks and challenges you've encountered early on in forming this company. I mean, there's challenges in the development, the IP, the fundraising. What does that look like?
Well, it's real interesting because most of the genomics today is being used in the medical market. And the medical, historically, always been a mentality of publish or perish. So the doctors want to have their own techniques, they want to have something to support. So you have a lot of different competing techniques that are out there. So you don't see standardizing like you do in a lot of other industries. So when you look at, coming out of the wireless industry, when they lock on a standard, it's the best performing product, the best price generally wins out. When going back to talking about like mammograms, there's at least 7 different types of mammogram you can go have. So you look at this can't be efficient. So that's a challenge that you run into with medical because there's a lot of competing viewpoints, and a lot of it's driven by the government through CMS, which is Medicare and Medicaid, what they provide for reimbursement. And the insurance companies base their reimbursement a lot off of what the government approves. So the difficulty is it's hard for them to get everything perfect, and they're trying to do the best job they can. But what happens is hospitals start looking at, optimizing their reimbursement, which doesn't necessarily match up with optimizing patient outcomes. So those challenges, what we're trying to do at SimpleSeq is we're trying to get the cost paradigm for genomic sequencing down to a price that you can do this for everyone. Because what we tend to find is more information in any industry really helps provide more solutions and more data.
Right. Do you have reimbursement for this approach?
Well, there's reimbursement for different types of testing. And what we're doing is we're working on the front end so that we're providing actually the kits and the assays.
Got it.
So the nice thing is the people that we provide these to can do it much more cost effectively, and they can still get reimbursement, but also it can help drive down the cost of reimbursement.
Who do you sell these into? Are these labs or these doctors' offices?
The process that we're developing, and we're taking a process called targeted capture. It takes 35 steps to complete. It's very specific, so you need highly skilled personnel to do it. And we've worked it down to where it's 5 steps. So you can get
And less skilled or still highly skilled?
Very less skilled. Nice. You can do it room temperature, so you can bring this into any lab, and it provides a different price point. It cuts out a lot of barrier to entry, and the beauty of it is every single lab that does genomic testing can use the products that we're making.
Nice.
Yeah.
Nice. So has fundraising been a challenge?
Fundraising is always a challenge, and especially when you get into high tech because it's so new. People have gotten to genomics 6-7 years ago. They're like, well, it's been so slow moving, but now you're starting to see that inflection point where it's really gaining acceptance, and you've got a lot of direct-to-consumer type genomics that have done really well.
Now, are you in the development phase, or are you launching somewhat commercially?
No, we're actually in proof of concept.
Proof of concept.
So we know that we've already proven that it works. Now we're working on is validation.
Okay.
So we're still talking to investors quite a bit. We've got our seed seed investors in place, but there's a huge opportunity.
And how does the FDA look at this from a validation perspective and a study perspective? Is it a multi-center, X number of patients? How do you prove that with the FDA?
Now the beautiful part of this is under, it's clinical lab validation, so it's what they'll call CLIA validation.
Right.
So we've got CLIA validation for genomics today. It's not really governed by the FDA. We're actually being proactive in talking to the FDA up front because it will impact so much what they're doing. And we're seeing the FDA is being very proactive on genomics in terms of the drugs they're approving. So the nice thing is what we're doing doesn't necessarily have to go through FDA approval, but a lot of the labs that we'll provide to have FDA approval. So what they'll do is they'll just change their standard operating procedures to show that here's this newer procedure, it's simpler, it's easier. It'll be much cleaner to work
what's
through.
the endpoint you have to prove? What is it that if you had this test, what does it do that you're proving that would cause somebody to say we're going to buy more of these tests?
The nice thing is we generally, when you're looking at a new test or a new method, you want to compare it with the existing methods to make sure the results have equivalency. And if it's better, that's great.
So you have a predicate that you're comparing it to that's established.
Yeah.
And how much better than the predicate do you have to be?
You've got to be equivalent.
Just, okay.
Yeah, because if you can provide it at a better cost, at a faster time, you're providing much better efficiencies for the same result.
Right, makes sense. So scientists are increasingly able to target the genetic cause of disease and repair it at the source. So can this approach stop disease in its tracks, in your estimation?
Without question. Because when you look at you're definitely going to the source. Because the source really becomes either a mutation or a tumor, or the tumors from a mutation. So you really can't get better information.
Right. And so from genetic research, are we talking about the possibility of developing curative drugs?
That's the goal. And the goal is to eliminate all cytotoxic drugs.
Is that what you're trying to also work in addition to the test, some curative drugs, or just we test it, we illuminate, and then someone else is going to work down the chain on the drug?
There's so many steps down the chain. Our goal is to get it to where the front end becomes much more affordable and faster so that you can develop solutions faster and better. So you got to kind of think of it as we're providing, almost like Intel, we're providing the chips that everyone else develops on top of.
Got it.
So we provide the raw material resource, but at a price point that really changes the game.
So it is estimated that genomic drugs will hit a $5 trillion addressable market, which is just massive. I mean, if I was an investor and you have a product that works and the industry is moving forward and developing, those are hard markets to find, right? This is huge. So when one thinks about genomic medicine, you're highlighting three interrelated but distinct technologies. One is gene therapy, where its goal is to replace a defective gene related to a specific disorder. Are these therapies durable, meaning a one-time treatment will address, or will you need to have multiple therapies when you think about gene therapy?
A lot of it, depending on when I tend to look at cancer a lot, because that's what we've obviously used genomics for the most.
Yeah.
They tend to be ongoing treatments because cancer is kind of an age-related problem. So when you say someone's predisposed to a disease, they may not have an issue until they're 60 or 70. And then that gene triggers and they start having an issue. So whatever you're doing to stop that gene from expressing itself, you're going to keep doing. The goal is to get to where you're using it, there's low-cost solutions.
So it's not a one-time, but sometimes you may have to take multiple bites at the apple to
it's going to be a lot. It's going to be a lot like a lot of treatments today, where the best is compared to like statins. So if you've got cholesterol, you're taking statins for the rest of your life, and don't want to digress too much, but a perfect example of when you're looking at sequencing, statins only work on about 60% of the population. So there's 40% that are just prescribed that doesn't work at all. In fact, it might even cause adverse effects. We can tell from the genomic markers who it's going to work on. And they don't use that today. Because they look at it and say, well, it's $10 a month, what's the problem? You should never be taking something that doesn't do anything for you.
Is it toxic, some of these drugs?
This is for...
or is it just benign, just not effective?
Some of them are benign, but anything that's going in your body's going to know what to do with it. So there are, adverse effects to every medication.
So it's only $10 a month. So I mean, if there's because to me, precision medicine's worth it. And if you can be more precise in what you're doing and more intelligent, it seems to me that would be the normal course you'd want to take.
Exactly. When you look at the commercial on TV with all disclaimers, imagine eliminating all the disclaimers because you only prescribe it to who it works on.
Right. Exactly. So the second area of development is cell therapy, which you talked a little about, which is genetically engineered cancer therapy designed to better treat tumors in the body. How effective has cell therapy proven to be?
I can give you specific examples because a lot of it, the problem is when you look at it on a mass basis, the best example I can give you is the drug Keytruda. Keytruda was based on genomic information, not on location. Mm-hmm. So it can be used in multiple different indications of cancer. And it's, in 5 years, it'll be the most profitable drug ever produced.
Really?
Yeah.
Interesting.
And it's interesting because they've always avoided basing drugs on genomics because they're like, well, it'll limit the market. But it also opens up the market.
Right.
Because it doesn't just focus on lung cancer, it focuses on multiple applications.
Right.
So I think that's really what, when you look at where it's going, it's going to change medicine dramatically.
So the third area is gene editing, where you can replace a defective gene with a non-defective or insert a gene where there's not one presently there. How effective have the trials proved thus far with gene editing?
Well, gene editing, the nice thing is everything where you've seen so far has been it's basically CRISPR-9 based and good results. The nice thing is you're not seeing people trying to make, there's always fear that you're going to edit genes and make a superhuman or something else. But when you start thinking about all the applications, think about being able to basically tie a drug directly to a CRISPR and edit a gene going into the brain. So now you look at brain cancers that you have a hard time treating because of the blood-brain barrier,
right.
That now you can CRISPR the drugs directly into the DNA. So now you can deliver drugs new. And these are things that, the applications are limitless.
Could you potentially edit the genes of cardiovascular disease, the predisposition?
Potentially you could edit any gene. But that's where you look at the trials and everything you have to go through to really make sure that it's safe. And the difficulty is not necessarily in the first patient, it's what happens in 2 and 3 generations.
Right. So where do you see genomic medicine advancing and what impact will it have on precision medicine? Because I was at a panel, not part of the panel, but I was listening to a panel of the leading CEOs of hospitals locally. And their big mantra was precision medicine. If we could be more precise, keep the patient out of the ER, be more proactive on health and wellness and those sorts of things. So with genomic medicine, how do you see it making an impact on precision medicine?
That's really the only way. Precision medicine doesn't exist without genomics. Because what precision medicine is, it's really targeting whatever you're doing, whether it's wellness, whether it's treatment, whether it's ongoing care, you target it specifically for that individual. And to understand them, you have to have their genomic makeup. And then you start realizing, here's what works for them, here's what doesn't work for them. And not just from a drug standpoint, but from a vitamin standpoint, from a food standpoint. It's literally genomics ties into everything out there. Yeah.
Couldn't you have a genomics test where it says this is what you should be eating and this is what you should avoid for your specific body and
you bet. There literally are tests like that right now. What you really want to look at, when you look at the genomics, a lot of that is what they call an assay. It's an array assay. Mm-hmm. So it does a single pass over the DNA and it reports what it sees.
Is that a swab or is that a blood test?
It could be either.
Okay.
So, and when you're looking at sample, you always, if possible, blood is always better because it's more pure. Saliva is always going to have whatever else is in your mouth in it. So you're always going to have foreign matter involved. So blood is always a better test. And when you look at it, you always want to make sure you're getting NGS testing, which is next generation sequencing. Mm-hmm. Because the level of repeatability of that is 99%.
Which I'm going to talk was my next question. So according to the National Center for Biotechnology Information, over 50% greater clinical benefit is achieved when precision oncology uses next generation sequencing. So what is the exact clinical benefit being achieved, or help us understand what is that?
Well, the difference is there's people here genomic testing, and there's a lot of ancestry type testing where they look at your
23andMe,
kind of your germline and bloodline, and we look at a lot of that as kind of recreational genomics because it's great information to have. It's not really what you use clinically because they're using an array assay and they're doing a single pass and they're doing the best interpretation from what they've got. So you'll get different interpretations from different providers based on what they see. With next generation sequencing, you're doing a minimum of 20 to 30 passes over the DNA, and you're getting a signal, you'll get it reporting back on a different frequency. So you can set thresholds to know whether that's the correct reporting. So that's why I say you get a 99% repeatability with NGS, because you want that level of repeatability. So for cancer, you do 500 passes over the DNA.
Wow.
So you're really thorough, and that's what you really want.
So you're not missing anything.
You're not missing anything, you're not getting anything reported wrong.
Right.
So you want correct reporting, and that's real important. That's where the clinical benefit comes in.
Because that's actionable, because if you have incorrect information, you're treating the
You can
wrong thing.
yeah, you can do something, you can make a change based on incorrect information, and you definitely don't want to do that in medicine. When you're looking at it and you're saying, oh, guess what, I'm Irish, I'm Scottish. They both all came from the same place.
Well, this reminds me of Theranos, right? There's a bunch of BS, bullshit made up, right? She's a master marketer, but you're talking about blood tests that people were using for very serious conditions, including cancer, and they were lied to, and a lot of people died. As I listened to the documentary,
it's mind-boggling to me, because to get the kind of specificity that you really need to do the in-depth type stuff she was talking about, you have to be using next-generation sequencing. And she was trying to make it seem like everything she was doing was a mystery and it was all proprietary, yet she was doing standard blood tests at the end of the day.
It's remarkable that she was able to do what she did, knowing what I know about investors, knowing what I know about the process. And the only thing I think of, she went after old men that were very wealthy, very, I think it was Secretary of State under Ronald Reagan, Schultz, for example, Weinberger, for example. These are people with a lot of money, a lot of connections that have no clue about medical device, about quality systems, about any of that stuff. And yet it still is amazing to me that she was able to pull the wool over so many people's eyes, and something to me it was so obviously, it was never going to work. I worked with a company called Sendex down in San Diego, and we were doing point-of-care blood testing or blood gas analyzing bedside, right?
Yep.
And it was very difficult to get that thing to work consistently, but I know there were so many regulations and so many things that had to happen. But the idea of a finger prick of blood, and that was it? I mean, people are smoking a dope believing that stuff.
Yeah.
But yet they did, and she raised, I don't know, was it close to $1 billion?
Yeah,
That's insane.
900 million.
That's bananas. I mean, good for her, but I mean, she's toast, but I mean, not good for her because it hurt a lot of people, but it's just crazy.
When you brought up investors before, we look at the level of scrutiny we go through, and something like that hurts the whole market, because you could go to any strong scientist who would have refuted it. And when you look at, you read the book Bad Blood, or you look at the documentary, and you'll see that their Stanford scientists basically poo-pooed it and said, that's the same.
She avoided all the venture capitalists in medical device or testing or genomics or people that are in the know. She avoided them like the plague because they would have just shot it down.
Yeah, it's amazing. So, but it's, you just don't know how something like that happens. But it does kind of, when you look at genomics, you're like, you're making that kind of promise without mentioning genomics. It's impossible.
Impossible. So let's talk about the three buckets, right? You got the patient, you got the provider, you got the payer. Just walk us through the benefits of this kind of testing for the patient. Let's start with the patient first.
Well, the patient, it's great because they're going to get stuff that works on them. I mean, a simple example is with pharmacogenomics. It's a panel you can do today. It'll tell you which literally over-the-counter meds will work on you. So, I mean, I've got a close personal friend who was taking Advil and was having all sorts of problems with it, and realized he can't take NSAIDs. Mm-hmm. So he can only take Tylenol, I mean, in terms of for headaches, and that's not uncommon. And it's stuff like that where if you just did a pharmacogenomics panel on when you went and saw your doctor for the first time, things he prescribed, he'd be able to pinpoint things that he knew would effectively work on you specifically. And that's at a simple basic level. And it's the same thing, we've got groups who work with doing epigenetics. So they do epigenetics testing, and they can see from certain, called SNPs, so they're biomarkers. They'll look at certain SNPs that'll let you know whether you can assimilate certain types of folate. And folic acid is typically not assimilated by most people because it's synthesized. And so they can look at B vitamins, they can look at D vitamins, they can look at any supplements that you might possibly take and specify, because not all vitamins are created equal.
Can it tell the doctor which drugs are appropriate for given pathology, which are effective? Because your friend just said, I take it for headaches, I can't do NSAIDs. So does that mean a migrainer taking a triptan? Could this genomic testing help the doctor, guide the doctor in the better triptan for this patient?
Yes.
Wow.
And it's like I said, with statins as well, because there's a lot of people on statins that don't do any good.
Right.
Nobody's giving them alternatives. No one's saying, look, you really got to change your diet. You got to, there might be something else that works.
Yeah. And knowledge is power. The more you can get. So that's the patient. Let's talk about the provider, the doctor. Talk about some of the benefits of precision medicine for the audience. What does that mean exactly?
Well, the difficulty with early adoption is it's very difficult because when you look at gene and relationship, it's not a simple 1 + 2 = 3. It's all the interactions and how the genes work together. So you can't expect a doctor to be able to assimilate this and be able to use it, basically give recommendations that they know specifically. So that's where you're seeing more and more tools coming out that are really saying, here's what this means. And that's what the BRCA testing does. It says, here's the relationship, here's the risk for that person.
So it kind of gives a correlation, not that it equals causation, but you get somewhat of a correlation.
It gives the providers much better armed to address the patient, especially not only from a illness standpoint, but from a wellness standpoint. So it can provide all sorts of recommendations from food through surgery.
So have you guys developed models where it talks about cost of care, the efficiency of care, now that doctors have this information, armed with this information? How does it impact, interactions with the patients? Because in our world, my world, it's length of care, it's cost of care, it's how does it change the protocol and procedure a doctor uses or applies on a given patient? Can we make it more efficient? And certainly as payers are looking at it, you get these expensive drugs just, subjectively, you diagnose a headache, for example, they give them a drug that's, $8,000 a year on a whim that the doctor's making the right guess. So how does genomic testing impact the cost of care for the patient that these delivery systems experience?
No, I think what happens is the cost of care comes down to the specific product that you're offering. So they'll do specific analysis for whatever panel they're producing or whatnot. What we're doing on SimpleSeq is really trying to provide all the front end so that for every panel out there, we can drive the cost out of it. But what you hit on the key is when you look at the cost of care, it's not just the cost of return visits. It's also the cost of eliminating adverse effects. So when you look at clinical trials, a big part of what slows trials down and stops trials are adverse effects. Mm-hmm. So if you have a companion diagnostic, we say, okay, for each of these patients, we do a genetic test, and we see if they've got these 4 markers, they're not qualified because it'll cause an adverse effect. Mm-hmm. You basically eliminate the adverse effects in a trial. So the trial moves much faster. And now you've got a target specific to the clinical, to the companion diagnostic to know who we really should be treating with this. Mm. And it goes beyond just, everybody thinks of that and I think of chemo. It goes beyond any drug because you'll see genetic predisposition. And what we're trying to do with the panel that we've produced is it's looking at 500 genes, where a lot of the panels today, like the BRCA panel, looks at 26 genes.
How many genes in total could you look at if you had time and money? Or did you just pick the 500 that were most relevant?
We picked 500 that all have any relevance to medical. So anything that has ever touched medical relevance is 500. So you look at the human gene specific to the human being, there's about 20,000 that you'd really say these are specific, not all related to medical.
And when you define medical, that means cancer, something that could kill you or really impact your quality of life.
Anything that says any medical research on it, on any level, is contained within about 500 genes. Okay. Yeah.
That's a good target.
And it's about 70, when you look at the markers, it's over 7,000 biomarkers. Yeah. So it's a big number.
Huge. So in 2018, the market forecast for next generation sequencing was $839 million. It is forecast to go to $4.1 billion by 2022. That's almost a little over 2 years away. How many companies are vying for this market?
I couldn't even tell you. I mean, there's a lot of players in it, and that's really the interesting thing is that's just the oncology market. So that's the cancer market.
That market there is just the oncology.
That's just the oncology market. Wow. So when you start looking at all the other applications, that's really kind of a drop in the bucket.
How does AI play into this world?
It's interesting because when you start looking at genomics, it's a perfect application for using advanced algorithms. So AI comes in, but what you need to be able to show is you need to be able to show how it came up with the solutions because you're applying these on humans. So you're seeing where AI is, a lot of people are excited about it, but a lot of the deployment side are cautious because you can come up with a solution, but the AI needs to be able to go back and show here's how it explains it. So there's evolutionary algorithms, there's AI, there's deep learning, there's quantum computing. It's really just being able to process massive amounts of data faster and look at all the correlations. And I think AI can only help. It can only help advance the science.
So is yours a test that could be sold on the consumer-based market where they could take it at home and they can get the results and through telemedicine send it back to their doctor, or does it always be will it always be a test in the doctor's office?
No, I think the beauty of it is because of what we're doing, we're not doing a specific test other than like the cancer is the first one we've developed. It's a product within a process. So we've developed a process. And the beauty of the process is it can be used on anybody using NGS. So the fact that we've taken a 35-step process, reduced it down to 5 steps, and made it so you can treat it with a much simpler mode, now you're looking at the possibility of actually bringing this into a lab design test. So now each hospital can have their lab run the test directly. So direct consumer, I think what you'll see is a lot of those will migrate to next generation sequencing just because they want that level of expertise, that level of quality. This can be used on anything. So what we're doing at SimpleSeq can be used literally on anybody.
So you developed
a
a process. Right now you're targeting cancer and genomics related to that. What are some other buckets you could target beyond cancer?
We're targeting everybody. Cancer was, so what we did was our process.
I like that smile, by the way.
Well, the beauty of it is our process, you can plug into whatever you're doing today and it'll make it more efficient. If you want to have more information, use the assay we developed, which is proprietary assay of the 500 genes with that process. So if you're looking at, say you're
So
looking
you're not just limited to cancer, you can
not
open
at all.
it up.
That's the first assay we designed, custom assay. So we can do a number of custom assays. We've got a number of ideas in-house, but we're a small shop and we want to roll things out one at a time. But the beauty of it is we can run a very large business just on the front end process.
Right.
And it benefits everybody that touches it.
So let me go back to Marietta. What is it called? Marietta.
It's Marietta
Company.
Genomics.
How does that relate to SimpleSeq?
Well, we started with the city of Marietta. They gave us a space to build our lab, and they want to really kind of foster growth and innovation in the biotech medtech space out in Marietta. Mm-hmm. They've left a large parcel of land open specifically for development, which is why Kaiser built their first digital facility there, Loma Linda built their Rades. So you're seeing it's working very well in terms of attracting high-grade hospitals. And what we want to do is we built basically a genomic incubator. So companies that want to get into genomics will help with their sequencing, and with our advisory board, we can help with the legal, the patents, everything else they need, and really help develop the company. During this process, we kept trying to find ways to reduce the cost of sequencing, and we wound up basically building our own incubator company. And that's how SimpleSeq came about, because we found proprietary ways to build the front-end process and then custom panels that can be used by it.
Now, does Marietta own any of your company?
Not at all.
And what does your patent portfolio look like?
For SimpleSeq, we've got 2 patents, and one specific on the assay, one on the process. And we've got another patent in process.
So you have a provisional, some utility. Have they issued?
Provisional patents, yeah,
Okay.
on the first 2 process, on the process and the assay.
I mean, that's saying a lot because this looks like a flooded market. I mean, with the market size, there's a lot of entrants. And the fact that you guys can come up with something novel and unique, I mean, you haven't gotten a patent, but the belief that you're going to get one, that's pretty impressive.
No, we're very confident. And the thing is, the stuff that we're doing, I'm being very general about it, because when you look at it, we've talked with a lot of high-end scientists. We know some of the top minds in genomics, and they're like, why hasn't somebody done this? So we know it works. We've already done the proof of concept. It's just a matter right now of really kind of tweaking it. And the partners we're working with, they're pretty big players in the industry that are working with us on the reagents and actually creating the product.
Do you foresee any kind of partnership relationships as you think about more mainline commercialization down the road?
Without question.
Yeah.
Yeah, because we don't, everybody's like, well, is your lab going to run this? And no, our lab's not going to. We can run stuff ourselves, but we want to provide this as open as possible so it benefits everybody.
So create a horse race, get everybody involved, selling it,
You
flooding
bet.
the market.
And it's going to help. It's really going to help the discovery, because when you look at genomic testing for now, a lot of people that have been tested for cancer have either been through charitable organizations or because they're wealthy. So you don't have a broad patient profile. So
Is this something you can
that'll
work
change.
with City of Hope with?
Without question. I mean, with every center out there that's doing cancer, it's a perfect thing to work directly with them.
And none of those organizations are doing what you're doing? That you're aware of?
They're doing next generation sequencing, but they're not using the process that we use.
And the secret sauce is in the process.
Yeah,
Drives the cost down and whatnot. Interesting.
yeah. And this is just, it's what we're doing is really riding off the work that all these others have done. Because you can't patent biology.
Right.
So we can patent the process because, they've driven a lot of research and a lot of work at billions of dollars that we've been able to leverage.
So your product pipeline, patenting this process, are there other processes or other discoveries that you could turn into other product lines or you could turn into other patents?
Yeah, we don't really see that much change on the process because it's at a point where it's pretty cost reduced, and for someone else to come in and try to do this, it's almost easier just to buy our product because it's not worth the cost of development, and you're not going to reduce it much beyond the steps it's reduced to that we're working on right now. Mm-hmm. And then when you start looking at the actual assays, that's where there's a lot of specific products we can roll out. But what we're really trying to do is we're trying to get people to look at as much of the genes as they possibly can because that's where the discovery is going to occur. There's things occurring within biomarkers that we have no idea today. And you look at, we're really kind of in the infancy in terms of what we've been treating. We've been treating cancer with precision medicine probably 10 years, and every year it's getting better and better.
We're kind of taking a sledgehammer approach to it right now.
We really are.
Yeah.
Yeah. So it'll be much more finessed.
Interesting. So your first product offering is liquid biopsy. What is that exactly?
Well, liquid biopsy is actually the second. The cancer panel is the first,
okay.
But liquid biopsy becomes critical, and that's where I was talking about ctDNA. Because when you sequence the cancer patients, you sequence them once for DNA because that's not going to change. The liquid biopsy really becomes your surveillance tool. When you start talking about wellness, today we do liquid biopsies primarily on cancer patients because then you can see, what their tumor was, and you can see circulating tumor in the blood. And then you can get proactive on the treatment. And that's when you talk about radiation therapy. If you're getting to it soon enough and you're keeping someone from doing more chemo or radiation, you're definitely helping their immune system. And that's really what you want to be doing. So then you can address it sooner. And we see with every form of cancer, if you catch it early, the survivability is very high. It's in the 90% almost across the board.
Fascinating. So I think about cancer, then I think about neuropathologies like Alzheimer's, dementia. Is this playing that world as well?
Well, there's definitely be biomarkers for neuropathology. And it really becomes, and that's one of the goals of our trying to drive more research and more study, and where you've got more patients where they look at it as like taking a temperature today. So when you go in, instead of doing EKG, they do an EKG when they also take a small blood sample and they run your DNA. Mm-hmm. And they do a circulating tumor because now, once you start finding more biomarkers, there's more things you can look for.
Is this true of every doctor's office visit I would go to? They take a blood panel and that's part of it?
Take it a step further. Every baby at birth should have a DNA sequence because that won't change.
Right.
And you do it once and you do 500 genes, you've got enough information for the life for that person.
So you know where the predisposition.
You know what the predisposition is, and then we see other things, you know what you can eliminate. And then you can really focus on where the real potential problems are, really looking at the core. Because a lot of what you do, you go to the doctor today, first thing they do is they ask you all their symptoms. So they're treating your symptoms. And not that that's their goal. They look at it and say, okay, the core problem for this is probably based in this organ. Here's the problem, let's address it. But in many cases, it winds up being you're treating the symptoms. Where if you look at even deeper, you start looking at, okay, is there a predisposition? Is there something that's wrong that could be systemic, or is it just a virus? Not just, is it a virus or is it a cold? And then you can address it better, and it's more information, it can only help.
Root cause. So it seems to me with all that information, it'd be good to have some AI or way to assimilate and process, because part of the challenge with doctors in like EKG or EEGs or whatever, they have to then interpret, they have to be the AI engine based on their Gestalt experience to understand what it's telling them, but even that's not foolproof. So the idea of what you're producing and all the data, and it could apply to so many different organs, and it could mean so many different things, which requires so many different types of disciplines to understand. it seems like this is a perfect storm for an AI that can help the doctor better process and understand.
It really is, and like I said, AI might not be the ultimate solution. We worked with a group that does evolutionary algorithms.
Yeah, evolutionary computation, whatnot.
Yeah, and they work very well because they battle each other and they come up with, here's the 5 things to look at.
Right.
Because it's typically not linear. It's not 1 or 2 things, and that's why the combination therapy works. So, all the deep learning methods, whether it's AI, like I said, quantum computing's getting a lot of talk right now. All of those can only help because they can take mass amounts of data and look at the correlations and start looking to see, okay, what are the interrelationships? But you're right. For the doctor, he needs to have a start to finish solution. It needs to provide him a report and say, here's how you treat it.
You need a happy meal almost because there's a lot of stuff going on.
You really do. You need to
reduce the complex to the simple.
Yeah, it's got to be an end-to-end solution. It's got to say, okay, we saw these, and it may not incorporate everything, but it can incorporate what we know is actionable and say, yeah, change this. And like I said, even down to the vitamins. No, they need a methyl tetrahydrofolate. That's the folate they need. They need they need more D vitamin, and this is what they can assimilate. It'll start giving them that specific information, even just for wellness, and for when you start looking at actual healthcare, it can definitely help in terms of here's how you best address this malady.
Well, what's cool about precision medicine, it's sort of like flying ahead of the aircraft. You can predict things before they happen, and you can almost allocate the care you're going to give something like that, and the probabilities that it will happen at a certain point in time. I mean, that's the whole beautiful thing about big data analytics. You can start testing these people for age, ethnicity, race, whatever, sex, and start to really identify and develop some very clear pictures. This person based on this population, you're in the 95th percentile. And you could be a lot more proactive, because right now I think we're all just reactive in medicine. And I think a big part of the goal, at least in my world, is prevent these guys from going to the ER because of the cost. But it seems like all medicine is reactive, and if you can make it more proactive based on wellness, lifestyle, understand the genetic components, predictive analytics, understand where this is going to go and when, I mean, that's going to foster a lot of other technologies and companies, I would think.
No, and it does. And it's funny because you think you're like, well, if you eat right, okay, what's right? It's different for each person.
Yeah.
And you exercise more, okay, different people react to different forms of exercise. So it's not as simple as, here's guidelines to follow. It does wind up being very precision and tailored to you.
Yeah, it's like the government. They're a bunch of boneheads. You think about the food pyramid, and what is that? I mean, it's like, eat this, and a lot of that stuff is just, I think as time has gone on, is not the best, but the idea of one size fits all is bananas, and it really has to be customized precision for the person, for the genetics, for their lifestyle, for their body type. I mean, not everybody can do high impact workouts, right?
Exactly.
It's just going to break them down.
Yeah, and it's and you're hitting on it because you look at it, and the government had all the best intentions in mind with the food pyramid and putting fluoride in toothpaste and stuff like that because they thought it was, well, this helps. Not everybody, and to an extent. So it does become real important that you know what's best for you.
Yeah, it reminds me of politicians. I mean, there's some good people, but there's a lot of morons up on the hill. I mean, when I listen to these guys talk, it's like, wow. It's just really truthful. I mean, we're not getting the best and brightest. And I think I like to think I work with a lot of really smart, thoughtful people. And so I think about the government, they would benefit more from connecting with private industry, certainly driving funding, and really get data rich on a lot of things we're talking about, because sometimes it feels like it's politically driven, who gets what money, when. There's a lot of industries that, from an economic perspective, you can't afford to let go, but probably aren't the better for the long term. That's all I'm saying, and I think the same thing's true in medicine.
No, I'll avoid politics just because they all drive me crazy as well. And what I do like is when you hear someone talking about bipartisan, it's like, that's called a win-win in business.
Right.
When both parties benefit. So there should be nothing other than that, because everyone has to benefit.
Which goes back to what I said, they're a bunch of morons that can't work together. Like, really? Is it that difficult?
Yeah, and but you hit it, I mean, because you do see that a lot in medicine too, where it's like, well, I helped develop this solution, so this is the one I'm going to use and promote.
Yeah, what is that?
And guess what? There's inventions every day.
Right.
There's improvements of progression.
Right. And that should be fostered, is what I'm saying. And I think you're right. I think
It really is.
If people could be bipartisan, I'd give them a lot of due respect. That's not happening. So it makes me wonder about the mentality, their emotional maturity. And that's what I'm saying, because I can see kids working better together, right? They got more wisdom in that process. So, because we're talking medicine, we're talking the biggest funder of medicine is the government. Right. And to get this right really does matter, as opposed to the politics and the jockeying for position and the grandstanding that you oftentimes see.
See, and that's the beauty of genomics. A lot of that can occur that's unregulated.
Right.
So a lot of the wellness stuff, that can occur without interference from outside sources.
Right.
And that's where I think a lot of what stops things in medicine is kind of the procedures. They look at clinical trials, they look at the cost. A lot of that can be circumvented by really taking a much better scientific approach and actually incorporating mathematics. And that's where you talk about AI, advanced, any advanced computing. It's using mathematics. In medical industry, they don't really use mathematics, they use statistics.
Right. No, that's a good point. And I think a lot of devices that are in the hospital or doctor's offices, it's a part of a solution. It's a piece of the picture. I mean, I was in another company, and I was selling a continuous cardiac output monitor. And I always tell the nurses, I said, you don't want to look at this number, CCO we called it. You want to look at the full picture. You want to look at all the variables, blood pressure, heart rate, temperature, all that stuff, right? And so it's really important to take in all of it to make an assessment, because it's not just one thing that's going to tell you a specific thing about the patient, because it's multifactorial, right?
Yeah. Well, it's funny because when you look at that, genomics gives you great input, but it doesn't, all the correlations have to come from the output. So you have to have all the patients saying what's happening. And when we were talking about AI, I looked at a study that was done using evolutionary algorithms, and they had blood pressure regular monitoring on patients. And the funny thing was, the patients that were distressed had better information because the nurses were going in there and taking it firsthand. So they would take it as it occurred. So you hit it right on the head. You've got to look at everything, and you have to have a system. This is where computing becomes so important, that can pull it all together and be unbiased, sort it, and come back and say, here's your 5 biggest issues. And there's no bias in it. Where if you have a human component, it's going to be biased, and it's never linear.
Exactly. Well said. I mean, that's spot on. I go back to the hospitals and I see all this equipment, and the doctor, a neurosurgeon, has a person with a traumatic brain injury trying to assess a bunch of stuff, and she's getting a lot of different data points on this patient. And you then have to, in your mind, be able to interpret where this patient may go. And you have to have an intervention, take them back to the OR, remove a blood clot, whatever. And it's really difficult because they got so many data points that they're processing and they're making bets on. And the idea of bringing it together under maybe a different method through mathematics, computer. I've talked to a lot of these neurosurgeons. They would love to have something like that, but they do the best they can with what they've got. Right. But the same thing's true of medicine at the family practice level. A lot of it's a guessing game, and we think it's this, and let's try this. It's sort of fix and chase and throw spaghetti up in the wall and see what sticks.
Oh, it's difficult. And that's where, and some of the best people I've ever worked with have been the doctors I've worked in the last 12 years. I mean, just human beings, they care, they want, and they're very accepting. So they're open to new ideas and new treatments. And that's where it's got to be, like I said, genomics can start driving that, but you got to have an end-to-end solution. Right. Because they don't have time for more interpretation. You got doctors seeing patients every 15 minutes.
You nailed it. That is such a key point. I mean, they got their protocol, they got their process, and they got their time in their day, which isn't a lot, right? Yeah. And the last thing you need is a technology that comes in and totally disrupts their process, adds another 10 minutes to a better test, or 15 minutes. I mean, it's got to be fit within their procedure, within their process, end-to-end solution, so they can look at it and it does the interpretation, and then they can make a call, like, we're going to go here or here.
No, and I look at healthcare and I hear all this debate about, complaints about the underserved population, you can't serve everybody. The thing is, we have to have a healthcare system that provides care to everyone, because when you look at the plagues and the viruses they've come from the poor because they don't have the best resource. They might be drinking water out of a stream or out of an unfit place. And that's where you're seeing things happening, such like AIDS. And that's where you need to be able to see the whole spectrum. You've got to treat the whole population so you're in front of it, right, instead of being behind it.
Well, it's like Bill Gates' malaria, a simple net to prevent people from getting stung or bit, Made all the difference in the world. One little thing, right? And you're right, because it's a big ripple effect across the world's populations that we ultimately have to deal with.
Yeah, and and that's where you just can't ignore a segment, and that's why when we look at genomics, we want to make sure that's a big part of what we were trying to drive the cost out, so that everyone could get sequenced.
When you say drive the cost out, what does that look like? It went from what to what? Give me some perspective.
Well, the steps, if you're doing targeted capture, standard targeted capture, it's a 3-day process. With SimpleSeq's process, with the process that we use, it's a 1-day it's an 8-hour process. So
what does that mean, 3-day process? A person somewhere for 3 days?
No, your sample somewhere.
So the sample goes and it takes 3 days of process.
Yeah, so if
you've got down to 24
so
hours.
if you send in something to like Foundation Medicine, which does most of the cancer testing, or Guardant, if they can get results in a month, that's fast. And I literally had a friend who was waiting for results from a cancer panel that he had done. And in the 6 weeks it took him to get it, he went from being treatable to not being treatable.
That's terrible. I was going to say, not only the cycle, but the actual physical manifestation of not getting on it quick enough. So you're in 24 hours.
Yeah, and you don't know that the treatment would have changed the outcome, but at least you have a chance.
Yeah.
And it's one where, and that's what that's what these cancer centers, they're busting their butt to get this turned around, but they got to collect a number of samples, because you can't just put one,
You can do yours in 24 hours.
Yeah.
That's huge.
You can do the front end, the prep, and then you put it on the machine. It's going to be on a machine anywhere from 8 to 12 hours, but you're still looking at a day to be able to run the test. And usually what happens is you're not going to run one sample on a machine. You usually want to optimize it and have at least 90 samples. So that's what happens is, if you're running this as a business, you got to get enough samples. But if you can cut the cost down and reduce the cost of that assay, you're going to get more people coming to the table. You're going to have better throughput. You're going to have better turnaround, and then you'll see results. And where you talk about the companies in the space, the majority of the companies are on that end of it, trying to come up with the application. I want to have the killer app. Right. So we're agnostic. We want to get them the data, because we're not smart enough to build all the apps. Get it out there where people can, who really have expertise in an area, are going to see, well, no, we've noticed that these 4 markers were up before, now there's 6 more.
Yeah, that's cool. So what impact do you hope your company makes in genomic medicine?
We hope that we really make it mainstream medicine. We really hope that it starts taking genomics from a specialized treatment to mainstream, where it becomes literally a screening tool. And that's the ultimate use, is where you use it for screening up front.
So what are one or two pieces of advice you could give budding entrepreneurs or CEOs on their journey, that if they took your advice, it'd be beneficial?
Don't stop exploring. Don't stop, don't get discouraged. It's funny, because we stumbled into SimpleSeq. So we were running the incubator, and we stumbled into a need by trying to improve a process that we're doing for our incubator companies. So it's, be very open, keep pushing, and just don't get discouraged. You hit on the biggest point, is the investors. When I talk to people, because I came out of the wireless industry, so I talk to wireless investors a lot, and they go, well, genomics, yeah, we really love the space. It looks like it's a booming space, but, I don't know, you guys are a small company. How can you come up with such a game changer? I'm like, well, Qualcomm did it to you guys. Right. And they're like, okay, point taken. And it's one where they look at it, and they kind of look at it like blockchain. Mm-hmm. They know it's going to boom, but they don't know which horse to pick.
It's sort of enigma wrapped in a mystery. It's complex. It's not 1 + 1 = 2, and I got it, and I can invest in it, and it goes to here.
Yeah, so it's really, you keep, and with us, it's like, well, look, everyone who uses genomics can use us. So for entrepreneurs, it's like, keep getting your story out there. Don't get discouraged, because you never know who it's going to resonate with. Right. And when someone gets it, they're going to get it. It's going to click. So you can't be discouraged, and you just got to keep driving and know that you're doing the right things.
Yeah, I've heard that startups are, it's a 10-year overnight success.
Yeah, well, you got to be honest with yourself first and foremost. You got to know that, okay, this is going to work or this isn't going to work. And you can't have blinders on. Right. And that's really the big difference. And that's really, because that's the advice we give to the incubator companies. A fast fail is not a bad thing.
It's a good thing.
It really is a good thing.
Absolutely. Yeah, I like your comment on blinders. So is there anything that you want to tell the audience we haven't covered?
No, I mean, obviously, when I look at SimpleSeq, it's one that, when you look from genomics, it's great play because it's used in every aspect. But the main thing is when you look at genomics, we've talked about humans this whole time. Plants, animals, air, water. And you're like, but there's no, water and air is not alive, but all the organisms in it are. So you see a lot of, I mean, agriculture is so far ahead of what we're doing with humans right now. You see them sampling air over their farms constantly to see what's in the air. And same thing with water. We talk with water purification of, okay, we can monitor what's going in and what's going out because we know you're going to get rid of all the solids. The question is, what microbes do you want to get rid of? Right. And what do you got to do to get rid of them? Right. So it impacts everything. So when you look at, you know, I talked with Chris Galvin, he was a CEO of Motorola when I was at Motorola. His actually dad, Bob, was the CEO when I started there, and Chris took over. And he was quoted back in Wall Street Journal back in 1996. They were asking about biotech. And he said, he goes, electricity was the invention of the century, of the current century. Biotech is going to be the technology, genomics is going to be the invention for the next century. And I totally believe that. And he said this in 96. So he was way ahead of the curve, but that's when all the genomic testing was really beginning.
Yeah. So I saw this interesting thing about low Earth orbit and the International Space Station, and they're looking at, you know, here on Earth you can print an ear out of tissue, and you know they're trying to print vasculature in the heart and major organs. And in low Earth orbit, since you don't have gravity, it's much easier to keep the striations and the lines of demarcations of the vasculature in the different layers where it needs to be. So do you see any genomic work being done at the International Space Station? Because microgravity is wholly different than gravity here on the Earth. It's big gravity well.
I'm laughing because we spoke with a woman that's really promoting that big time and is really into it. And I know that there's people doing 3D printing of organs on Earth. So I don't put...
They're also doing it up there.
Oh, I know. And that's what I mean. When you say, do I see applications? It's so funny because it changes the condition. It's like running servers underwater. Right. It's things you never thought of that people are doing. So no, I don't eliminate any possibility. I mean, I just think it's great.
Yeah.
Because you know, I think I'm pretty open-minded, but I hear stuff that just blows me away. I'm like, I never thought that would happen.
Yeah, it's cool.
So it really is.
Yeah. John, thanks for coming on. Enjoyed it.
It's been a pleasure. Yeah. Thank you, Ben.
Thanks.